Welcome. I just want to quickly review about viruses because I like to say they're smaller than smoke and very ancient and infectious particles. They can be naked or enveloped, have DNA or RNA, and they are just little parasites that have evolved very quickly and over long periods of time to get into every kind of host imaginable and usurp its machinery to make copies of themselves. I like to think of a bulldozer running into a car factory and running amok and creating new machinery to make more bulldozers, and then they blast out and go into other factories. So with that said, and just want to reiterate, viruses, they do get into specific hosts and typically specific tissue types and cells. They can get in mostly where our defenses are a little bit weaker, where there's mucus like the respiratory system or even the conjunctiva of the eye, and then into the intestinal tract, or if the skin is damaged in any way, a small scratch. But even some viruses have figured out how to just get in through the skin. And then, of course, we talked about arbor viruses, arthropod viruses, which mosquitoes and flies and ticks and so on, which are very effective transmitters. We do have a lot of defenses, and I like to envision them as different walls that the virus has to get over. And our anatomical defenses are considerable. Mucus not only traps things, but we expel it all the time. Saliva, tears in our eyes, which are not only viscous, but they have enzymes in them. It's amazing we don't get more infections through the eyes, but they can. That's why so much advising about not touching your eyes. Scabs and any kind of minor wound. It's the same with bacteria. We do have an intrinsic and immediate response. Now, I don't want to go into all of these. These are a little bit complicated, but we are going to talk about some of them. And then we have a remarkable system, mostly born in the blood and also born in the lymphatic system, which you may not be that familiar with, and we'll go into some detail. It's unfortunately a pretty microscopic and detailed system, but I'll try to make it as simple as possible. And we won't go through every step of it, but we do have this innate immunity that it resists lots of things. It's amazing. And all it takes is when something dies, the immunity is gone, and all of these things, fungus, bacteria, viruses, absolutely worms, just take over and devour the corpse in a very speedy fashion. So without that immune system, we are decomposed very, very quickly. We also have a very important acquired immunity. It's a little slower because the white blood cells have to figure out what's going on, what is in us, what do they need to defend against. And these are two different types of white blood cells. And I'll talk about those because they're extremely important, especially the B cells. And I know you probably haven't heard of these, and you'll probably never see them, but they are there. So our immunity is dependent upon a lot of things. The chemical nature of the skin, it's oily, it's thick, the outer side is dead, so it's hard for any virus to get in. Usually it's some sort of insult, and it's the same for bacteria or fungus, some sort of scrape or insect bite. The GI tract is loaded with acids. And we have, very importantly, our general health that supports this immunity. I'll talk a little bit later that we really can't boost our immune system other than staying healthy, which sounds counterintuitive, or it should be intuitive. The white blood cell production, there's nothing we can do to enhance that except when we do get sick. And then there's a chemical system, and unfortunately it has a number of names, but white blood cells are also called leukocytes. And so white blood cells talk to each other through chemicals called interleukins between the white blood cells. There's also a chemical class called cytokines. You'll hear a lot about that because this is what is the lethal part, because sometimes our body cannot get on top of the infection. Fortunately, when we make antibodies, we keep a kind of memory. Some of the cells retain what they did in previous infections, and we call those memory cells. All right, let's see, oops. There, I just give you this, there is no way to build or boost your immune system. Many people have made fortunes off of this. Outside of just eating healthy, fruits, vegetables, little meat, little fats, good diet, exercise. Exercise, in a way, especially good exercise, will kind of fool your body into thinking it's attacked by a fever. And so it will actually react as if you were doing damage to your body. And I was just reading last night about how we think testosterone increases during exercise. Testosterone increases when you're watching exercise, not when you're doing it. It lowers it, but it does increase the immune system because, again, you stressed your body. Avoid stress. Stay hydrated, sleep well. Good advice for anyone. Taking vitamin supplements, that's fine. None of these micronutrients or the special new things like turmeric or other things have not been demonstrated to do anything to the immune system. You're just making some companies rich, and you can overdose. So we have not only physical defenses, but we also have chemical defenses. Saliva has enzymes in it. Our stomach acid, urine is very toxic, flushes things out. The mucus, and I'll talk about interferons, which are remarkable things. This system requires another system that we're not familiar with. We have a circulatory system, but there's a second circulatory system. That is the lymphatic system. And the lymphatic system is made of a parallel veins, if you will, and these go all over the body, and they pump fluid, or you pump the fluid, basically plasma, not the blood cells in most cases, certainly not the red blood cells, but they do pump white blood cells and plasma, and pumping is done by movement, which is also important, that you get the exercise to pump it. That's why your feet and legs swell sometimes if you're not walking, because the muscular contraction moves the fluid up through the body. And periodically there are nodes in the armpit, nodes in the neck, nodes in the inguinal region, and nodes associated with the intestinal tract. And these nodes are clearinghouses. Some white blood cells develop here, but when a white blood cell is cruising through the blood and it comes in contact with a virus, it will try to eat it if it's a particular type, and then it will bring it back to the lymph node, which sounds very strange, but it then interrogates it, if you will, shows it to its other white blood cell brothers, and often kills it there. So this is one of the reasons the doctor palpates your nodes, because they will swell if you're fighting infection. The nodes behind your neck, underneath your chin, and so on. And of course, certain cancers will travel around through this system. So there is, parallel to our bloodstream, another set of tubes where the plasma will leak out and travel and then dump back into the bloodstream up here underneath your collarbone. There are a couple tissues associated with this, or organs: the spleen, which is mostly a sort of a reservoir for blood to pool, and the thymus, which is just underneath your breastbone. Some people eat it from animals. The tonsils, the adenoids, and so on are also part of this, and a whole bunch of patches in the intestinal system. So if we look at all the nodes, you have them behind your knees, up in the armpit, all the way through the head, lots of them. And unfortunately, a lot of them around the breast tissue, because this is how fluid gets in there, but also how the breast tissue gets drained. And so cancers will travel through these nodes, and that's why it's important sometimes in surgery to remove those. Now, if we take a really thin tube of blood and spin it, centrifugate it, we will show that about 45% of what you think of blood is red blood cells. The rest, almost 55%, is plasma. And this is not a clear fluid, but relatively cloudy, yellowish in color. And I'll tell you what all of this contains. It contains lots and lots of stuff, and it is the plasma that moves through the lymph. Then you'll get this little, what is called a buffy coat. And this is less than 1% of this volume, but it contains all the white blood cells and the platelets, which are important for clotting. Now, other things found in the plasma will include electrolytes, sodium, potassium, glucose, and proteins, hormones, that's how the hormones travel, waste products, that's how the urine gets formed, and what we call proteins. And many of these are clotting proteins, but also albumins, which are generalized proteins, and immunoglobulins. And these are the basic constructors of antibodies. And we'll talk about that just a minute. Now, if you take the blood clotting factors out of plasma, you basically have serum. And serum, you can sell plasma. It's very expensive. I like to tell my students, I bought my first hematology text by selling my plasma. I thought it was a fair balance. And I got $15 for it at that time. Unfortunately, it couldn't pay for the whole book. But if you do a stain of your blood, you stain it with a particular kind of stain, you will see your red blood cells, which are kind of empty looking, like a lifesaver. They don't have a nucleus. They're short-lived, and there are a lot of them. Then you have various white blood cells, and you have a giant one with a big nucleus, which we call a monocyte. There are smaller ones with big nucleus as well. These are lymphocytes. And then there's a multinucleated one, which we call a neutrophil, a basophil, which responds to inflammation. Eosinophils mostly respond to allergies. So if you see something like, or if you have a respiratory problem, they do a blood test and they see a lot of these, you probably have an allergy, or if you have a lot of these two, you probably have a worm or a parasite of some sort. So the relative proportion of these is very important in figuring out what is going on with the body. Now, our first line of defense are these big, big monocytes, and they turn into functionally amoebas. They are amoeboid. They can identify things that shouldn't be in you, and they have the ability to engulf them. So they become macrophages, big eaters. Then we also have a cell, basically a type of white blood cell we call the natural killer. And it is also able to engulf or convince the cell that's infected to kill itself, self-destruct. They also regulate how much interleukin is produced. And neutrophils also can do this, depending on how they encounter everything. Then there's another cell which travels a lot in the lymphatic system called a dendrocyte. All of these are important because they're able to identify things that aren't necessarily supposed to be in your body, and they don't need supervision. Dendritic cells look really strange. They look like amoebas, and they're able also, by acting like an amoeba, to just literally engulf whatever they find. The most important thing they do is they can dump enzymes on it, break it apart, and then take a little tiny piece of it and present it on the outer surface. And this presentation is really, really crucial. They're not the only cell that does this, but they will go back into the lymph system and say, look what I found. And I've never eaten this thing before, and I want you to be aware of it. So they present it on a piece of protein. And what they're presenting basically is the antigen that they have discovered in this virus or bacteria. In addition, since they know they've encountered something, they begin to release cytokines or interleukins. Interleukin, I think, is a better word because it tells you they're communicating almost hormonally between white blood cells. When these interleukins, cytokines are produced, it activates natural killer cells and what we call T cells. And the T cells turn into two different things. One, there's a helper cell, and then this turns into a killer, a cell-killing T cell. So specifically, here's a virus or bacteria. It's engulfed, literally swallowed. Enzymes, lysosome enzymes break it apart. Then they take the surface molecule and put it on one of their molecules, which is called the MHC. And I apologize for this, but this is the major histocompatibility complex. And every one of your major histocompatibility complexes are unique to you. And we have to match them if we're going to do any kind of organ transplant. We don't have to match them if we're doing blood transfusions, but we have to match them as closely as we can in organ transplants. What the macrophage or the dendrite will do is take this little antigenic part of the virus and then put it on its surface, and it presents that antigen, and it says, go find this. It's like giving a piece of something to a bloodhound and say, go find this, and when you find this, kill it. So it tells the T cell, which is now a helper cell, and the T helper is going to take this to the lymph node and tell all of its friends about this new invasive pathogen. It gets a little complicated when we go to the B cell, but it's the same process. Here's a macrophage or a dendrocyte eating whatever microbe. It breaks it apart. This is the antigen, and this is the specific antigen. It puts it on its major histocompatibility self protein, presents it to a T cell. And this T cell will then put it on its surface, and along with circulating chemicals, present it to a T cell, which will now become active killer. T cells generally are quiet, but once they are activated by antigen presenting dendrocyte or macrophage, they become killers. And the same thing happens, they'll prevent it to a B cell. Now the functions differ. Think of T and toxic. The T cell is able to kill anything that has this antigen on it. They're given carte blanche to look for that antigen and kill whatever is presenting it. The B cell now takes that antigen and begins to make specific antibodies against that antigen. So I love this because this is actual computer generated image of a white blood cell, a macrophage. This is what amoebas look like, and eating a series of cockle bacteria. You couldn't take a picture like this and show them eating viruses. They're just too small. And interestingly, about the body, if you show people who are sick a lot of images of white blood cells, especially macrophages, and tell them about them, this is one way of actually increasing your white blood cell count. Visualization somehow makes you produce more of them. You don't need boosters, you need education. Inside the lymph node, and this lymph node has a lot of blood as well as lymphatic tissue going to it. A white blood cell, like a dendrocyte or macrophage, comes back to the node. That's where the presentation to the T cell occurs. So T cells are in the outer part of this node. And then the B cells are activated. They're in the what is called the medulla, the middle part of this node. All of these are lymph ducts that are coming into the node. And then you have one going out. There are little one-way valves, just like the veins have, to prevent the lymph from going back into the body. This is very crucial also for when you're walking and pumping the lymph around your body. It prevents this from flowing back down into your legs or something like that. Now the interleukins, or the cytokines, which the press likes to use, are released by most of the white blood cells, and they signal to each other, and they have specific functions that help fight the infection. There are over 31 different kinds of interleukins, and they are generally just designated as IL, interleukin, and then numbered. And specific ones like, well, we'll talk about those, have specific roles. Some promote inflammation, which is a crucial part of fighting disease. Inflammation is a normal response to infection. It makes the body unpleasant for the virus or the bacteria. Sometimes it runs away and we have to try to take anti-inflammatories to protect ourselves. And that's where most things like aspirins and cortisones and so on work. They're anti-inflammatories. Some of the interleukins do that themselves. They inhibit the inflammation. They say, that's good, we got an upper hand, let's slow this down. And then some do a number of different things, and we don't have time to go into that. Some of these interleukins are designed just to go after tumors. So you'll see these tumor necrosing factors. And then probably the most intriguing, since all animals have been dealing with viruses for eons, they have developed a specific interleukin called interferon. And I'll talk more about that. So these interleukins regulate the response. Interferon is probably the most important. Some of them, after the infection, will cause the body to repair itself. You may need to scar tissue to form and so on. But the interferons are specifically made to react to viruses. You can see all these different kind of major interleukins and their target cell, the source cell, the target cell, and their biological activity. Don't worry about this. You're not going to be tested. I just want you to realize the complexity of this and how this thing can get out of hand. If you have an allergic response, then most of the interleukin will be IL-5, and they'll work on the eosinophils, and you'll have hopefully a controlled allergic reaction and not go out of hand. Sometimes this does go out of hand and you can get a severe shock like anaphylaxis, and it can be very quick. Well, let's keep going on, because we think of inflammation as a dangerous reaction, but it is an essential reaction. And it is important because it's a quick response to insult or infection. And it often becomes quite localized and acute. And it involves a number of different localized cells. Don't worry so much about all this detail, but there are some cells like basophils and other kinds of epithelial cells that release a chemical called histamine. And histamine will affect the blood vessels, so the blood becomes leaky and dilated, and it will rush more white blood cells to the area that is damaged. There are a couple of other things besides cytokines that are released. We don't have time to talk about the complement system, but this is a sequential system, and it affects every part of the body, especially the cytokine. And sometimes this is chronic. Some people have this over a long period of time. Most of this is an emergency response to some sort of infection, but sometimes this can be chronic, and cancer can cause this, neurological diseases, and autoimmune diseases, which are very, very strange. They can cause the body to have chronic inflammatory response. And sadly, most of these are females because of, well, the fact they have two X's and some other things, but things like lupus. Way back in the 1st century, Celsius described this, the inflammatory response, with four different words: rubor, dolor, calor, and tumor. And basically it refers to redness. That's the first thing that happens. When it gets inflamed, the blood comes and it swells and it gets red. The tumor is the swelling, the edema, and then the dolor is the pain, and then often there's heat, not just from the blood, but one of the interleukins goes to the brain and causes the body thermostat to be reset. There are other things that happen. You will not really feel like eating. There'll be clotting taking place, especially if you have some sort of damage to the skin or the blood vessels, and there'll be other sorts of actions. But inflammatory response, very quick and very important and generalized. The initial inflammatory response comes from interleukin 6 and also interleukin 1, and it goes to the brain, and it is a natural response. It's trying to make the body uncomfortable for the interloper. Bacteria are very temperature sensitive, and so if you can burn it out, and this has been demonstrated over the ages, viruses are less sensitive, except many of the naked RNA viruses are sensitive to increased body temperature. So it is not only a symptom, but it is also an attempt to deal with the pathogen. We take, so think about fever. It's a marginal increase in body temperature, but it is trying to kill whatever is there. Sometimes it does run away from us, and so we tend to try to take prostaglandins or antihistamines. Remember, a histamine is released by this endothelial cells, and histamines, as you know, will make it swollen. It will itch in some cases, especially just a small amount will cause runny eyes, itching skin, and so on. So you put either a cortisone or an antihistamine to deal with this, like Benadryl. Prostaglandins are a special type of anti-fever substance that we also use in our drugs. And aspirin is also capable of lowering the body temperature. So inflammation in general, the capillaries dilate, white blood cells then can sneak through the cells and get into the area of injury. So this dilation is important. It brings more white blood cells to the area. You'll get the redness, the heat, the tenderness, the pain, and then one of the interleukin 1 and 6 will get to the brain and cause fever. So all of these are an initial natural response. Now, you'll hear a lot about the fact that sometimes this interleukin cytokine system gets out of hand, and it can, simply because the viruses can grow faster than the body can deal with it. So the initial interferon, different types of interferons, the colony stimulating factor. Here's the interleukin 6, which is doing the fever, and then interleukin 8 and 1, which is also resetting the fever, tumor necrosis factor. All of these, if they run out of, get out of control, and they can't deal with the infection, you will get a whole bunch of things happening at the same time. The immune system will sort of paralyze itself. You'll have lung damage because the fluids, the lymph and the blood will flow backwards into the lungs. You'll get clotting in places you shouldn't have clotting, and you'll get shock and even kidney failure. So this is called a cytokine storm. And you have to be very careful and watch for this. Low blood pressure will show that the shock is imminent. You'll have a rapid tachycardic heartbeat and trouble breathing and high fever and so on. But this is normal. Well, the immune response is normal, and it's normally self-limiting, except in these severe respiratory cases. And it's very sad, but the lungs fill up with fluid and there's coagulation and so on. So the storm can begin later, even though you've sort of seem to have the first phase and the viral load decreases, but then the cytokine response runs away. So even after 7 or 14 days, you can still have a very acute crisis with a cytokine release. I know this gets complicated, but I just want you to be exposed to it. So we've talked about what's happening. And this is why these diseases are called severe combined acute respiratory distress, ARDS, which means that you're not getting any oxygen into your blood because the lungs, the alveoli of the lung are filled with lymphatic fluids and also blood. So the cytokine release, it's hard to reverse this. We have very few anti-inflammatories, except because you don't want to do it initially. Sometimes you can do massive cortisones, and prednisone type substances, which are potent steroids, to try to stop this in its track. But this is normal until it gets out of control. So remember, this is our first response, these natural killer cells, these macrophages, they release cytokines to stimulate the T cells and so on. There's also another marker that is done pretty quickly that comes out of something called the C-reactive protein. These are acute phase proteins that are present in the plasma. And it's one of our best indications that something is going on seriously. And so blood is taken, and you look for C-reactive proteins to see that there is some sort of major inflammatory response. Oops, going backwards, sorry. Now, the important thing, we've kind of covered this, the presentation. This is a macrophage, a dendrocyte, or a neutrophil that's eaten a virus. It's taken the marker, usually the surface marker of the protein of the virus, presents it on its own little surface major histocompatibility complex and presents it to the T cell. The T cell will make a helper, and the helper then stimulates B cells. And the crucial thing to remember about B cells is that B cells then look at this particular antigen and begin to make an antibody against it. So just remember, antibody is a response to this little antigen, and it is made by B cells. The T cell is now turned into another killer, a natural killer, or a macrophage. And I like to kind of, back in junior high, somebody would run up to you and pat you on the back, and there'd be a sign saying, kick me or hug me. Where I grew up, it was mostly eat me. So antibodies do this. These antibodies are little tags that are put onto pathogens like viruses or bacteria and tell the macrophage and the natural killer cells and everything else that is capable to eat it. Now the antigen, we briefly mentioned this, antigens are proteins that don't belong in our body. They may be combined with sugars and fats, and each one has a different shape, and they stimulate a complementary antibody response. And this can be found on pollen, it can be found on viruses, bacteria, worms, and so on. But the shape of this marker, the antigen, is crucial because the antibody has to be redesigned and basically chemically evolved in hours to match it, to match a little structure that they'll hold on to. And the antibodies are made by the white blood cell, the B cells. They're Y-shaped proteins. They're pretty standardized, except for this very tiny end. I like to think of these as lobsters. And the tips of the claws are designed specifically for a specific protein or antigen from a virus or pollen or whatever it is. And this is actually evolution at a cellular level that takes place within hours or days. And they have to run through, they have to make new ones every time they encounter some new antigen. So this is a de novo process. And once they figure it out, then that particular B cell continues to make this antigen to fit to this, or this antibody to fit to this antigen. We can't make, in most cases, we can't make antibodies, but what we can do is maybe make a form of this to inject into people so that the body will respond to make an antibody against it. And believe it or not, this is a strategy for immunization, for vaccination. If we can kill this and just leave this, or if we can just inject this, or if we can put this on something else that is harmless, we actually have a form of vaccination. Now, the antigens or the antibodies can assume a number of different shapes. Come on, am I waiting there? They can be single, and we do have sort of generalized ones like immunoglobulin. The IG refers to this is a kind of protein and designated as immunoglobulin. And G is the generalized one. So is A, but A is unique and it has two of them together. E is one that is functionally against allergies or pollens. And D is one we not quite sure all the things that it does. And sometimes these group up and are able to combine and inactivate a whole bunch of antigens. What their function in is mostly they stick to the virus and they prevent the virus from docking. So here is a regular virus ready, a rhinovirus ready to attach to its receptor. But if the antibodies are covering this, then it can't attach to a receptor and it basically can't infect us. Now, this is a great system and all sorts of things have it. I recently got to talk to one of my former students who's working on this system in fleas. And another system is working, or another student is working on this system in actually horseshoe crabs, because these immunoglobulins were developed very early and every animal has them, and they are important for how do we control them and how do we figure out how to develop our own. Now, if you take a whole bunch of antibodies from people's plasma and you basically winnow it down to functionally immunoglobulin G and A, you now have a functional immunoglobulin injection. It doesn't have all the immunoglobulins, but it has a generalized one. So sometimes we do this, we give this to a person who's likely contracted hepatitis or is under initial treatment with hepatitis. Now, we can't use it not strictly for most viruses, but in some cases, it gives us an additional time. So once a big eater or a dendrocyte has eaten this, presents the antigen to a T helper cell, it presents it to a B cell along with stimulation from interleukins. Now the B cell declones itself, it divides very quickly. And one of the cells becomes what we call a plasma cell. And all it does, once it figures out the structure of the antibody, makes massive quantities of antibodies that will flow through the lymphatic system and the blood system, trying to find representatives that have the antigen, that is the viruses and so on. But some of these B cells clone and become quiescent and become what we call memory cells. And every time they don't ever disappear. The plasma cells, once the infection is cleared, will disappear. The memory cells hold a genetic memory of this encounter. And hopefully, if we encounter this pathogen again, this memory cell will respond very, very quickly and clone itself to make new plasma cells that release that antibody. And this happens all the time. If we encountered something as a child, we have a memory cell of it. If we encounter it again later, the memory makes a plasma cell, it controls it, and we never even knew we were sick. Oops, I keep hitting the wrong one. Now, what do antibodies actually do? Well, they do a number of things. They can neutralize. They can just stick to the virus or the bacterium and they won't let it do anything. They can clump them. And this clumping or agglutination is good because then white blood cells find a whole bunch of them. They can precipitate them. They can cause them to come out of the plasma. And they also, well, we don't have time to talk about this. This is a secondary system. They can enhance what is called the complement system. And they enhance phagocytosis. And in some cases, that is eating. So once these are marked, remember, white blood cells now can identify that and have free rein to just ingest it and eat it. And in some cases, the complement can cause an infected cell to actually burst, which is known as lysis or cell death called apoptosis. So they can neutralize, they can cause them to clump. The opsonization means that they will be easier to ingest. They can cause the complement, which may lead to the lysis. The complement helps open pores, and this can enhance the natural killer cells and so on. So it's a very complicated system. And it isn't that quick. It's the quickness of our neutrophils and dendrocytes, and it takes a week or so for antibodies to build up. And many of you have heard that, well, if a COVID patient has recovered, then they should have managed to make specific immunoglobulin G antibodies against COVID. And if the person's ABO blood type matches, you can take a plasma donation from this person and put it into a recipient. And now that recipient has already activated immunoglobulin against COVID. This is very effective. Not many people do it. It's timely, it's costly, and so on. Now, of course, viruses have been at it for a long time, so they do a lot of things. They can inhibit antigens. They can remove certain classes of molecules that make the antigen. They can make decoys. They can influence the receptor of the cytokines. And they also very recently have been found that they can communicate. Viruses can talk to each other chemically. They have their own chemicals, and they can block both interferon action and also say, avoid this cell. This cell is filled with interferon, and it's not going to do you any good to try to infect it. So they keep looking for naive cells that haven't been infected and don't have interferon to block them. Now, how do we know if antibodies are present? Well, we functionally make an anti-antibody. There's two ways, actually. We can put a fluorescent marker on the antibody and then put it in people and then take the piece of blood out and then see if that fluorescent marker is present. The fluorescent marker, by the way, is mostly from jellyfish. So you never know where science is going to lead you. Most of the time, it's an antibody against the antibody that you're looking for that is marked with a fluorescent marker, immunofluorescent marker. This is the principal way we detect antibodies. Now, interferons are released as an interleukin, and it's unbelievable what they do. Not only do they activate natural killer cells, but they promote an enzyme in themselves and also especially in their neighboring cells. If a virus gets in and the virus is trying to reassemble itself, in the last gasp of the host cell, it will release interferon that will go to the neighbors, literally all the neighboring cells, and activate what is called protein kinase, R. So this protein kinase, once they get this signal, now resists viral infection and kill it. So a remarkable substance. And by the way, we have manufactured it genetically, biogenetically. We can manufacture different types of interferons. And it is given particularly in initial virus infections. So the body makes this very quickly, but it may not be made for long. So it's trying to slow down the initial viral load because it takes a while for the T cell response, where it begins to activate T cells into killers and activate B cells. Now, the macrophages and neutrophils and dendrocytes are active here, but interferon comes later. And it has not only an antiviral, it regulates the immune system. It can cause cell death if the interferon can resist have the neighboring cell resist. It can also cause this cell to blow up, and it can also increase blood flow and do a number of different things. We have made this through recombining. We cut the gene out of interferon and put it into a bacteria. We grow the bacteria in vats, and we then isolate the interferon and can use it. It's why it's called the recombinant interferon. Unfortunately, it's short-lived effectiveness. It is also used in cancer, anti-cancer treatments. Very wide-ranging effect. I don't want to talk about all these, but the main effect is this protein kinase, which will inhibit the virus RNA and actually kill it eventually. Now, viral infections, most viral infections are acute. That is, you have all of this response taking place, and then you clear it. The antibodies and the T cells and natural killers have done their job. The interleukins have done their job, and you have cleared. It seems like COVID is a self-limiting system. Unfortunately, there are a number, because of making proviruses, that you have a persistent infection, or I put this in because many of these are chronic and persistent, but you may have subsequent outbreaks, and you may not even know this. The herpes virus, in particular, hepatitis B, and some others. Persistent, a number of lymphatic leukemias or meningitises will be persistent and then suddenly take off for some reason and kill you. You can have latent infections like herpes, which will have another infection, another infection, another infection. All of these may mean cold sores or blisters or something else. And then there are slow viruses that just stay there without any indication and then suddenly come back. Measles, believe it or not, can kill later in life. And likewise, HIV, which you may be able to control, and then when it erupts later, you're going to die from it. So if we look at this, the virus comes in, you have the initial reaction with killer cells and dendritic cells and macrophages, but eventually you begin to make antibodies. The antibodies are produced. You increase the number of T8 and which are cytotoxic, and the T4 cells. So this may take 30 days for all this to happen. But the important thing is you may have, if you survive this long, you will produce memory B cells and memory T cells so that you have a quicker response if you ever get infected again. So the course of infection, your first exposure, you get antibodies, T cells. You may not You may have a second infection, but you're not even aware of this. And then you may have a major infection later, but you have these T and B cells that will respond. And you may not be aware of any of these later. It's important to know what the incubation period are, because you have to respond very quickly. So influenza, 1 or 2 day, and if you can't develop an immune response in that time, you're not going to do very, very well. I'll talk about this later. I think this is very well worked out. It's kind of old, but it shows a series of clinical features in the intestinal tract, the blood, and then eventually this central nervous system. If it makes it this far after about 2 weeks, then if paralysis is going to occur, that's when it usually occurs. Only about 10% of polio infections actually make it this far. But in the meantime, all of these in the intestinal tract, the blood, are shedding in the body. COVID does the same thing. The incubation is about 5 days. You won't really know anything until about 5 days with, say, loss of smell, cough, headache. Then you'll go through mild acute phase, and then you'll go through the severe phase after about 2 weeks, mostly characterized by common things like difficulty breathing, cough, fever, fatigue. So the initial response, which are the natural killers and the macrophages and dendritic cells, try to catch up with the virus, but the virus, if it keeps going, then you'll have this adaptive response of T cells turning into memory cells and cytotoxic cells, stimulating B cells and antibodies, and then B memory cells, which will be cloned and recloned and recloned. So a very complex system of different kinds of cells, different cytokines that will cause fever, impaired blood flow, decreased serum proteins, and liver damage, anemia, all sorts of other things. Here's the acute phase protein, which will respond, and that's what we look at to see where the infection is in its terms. Now, well, I don't need to go over this again, but basically, there are no such things as immunity boosters. It's a marketing strategy, and zinc, and I like this, the turmeric extract, that actually was copied right from an ad. They couldn't even spell it. And I'll just mention my law of expertise. The less we know about something, the more experts there will be. And we know very little about the immune system. And there's no way, all the research has shown, there is no way to stimulate it other than exercise. You can't enhance antibodies. You can't enhance white blood cells. We like to think we can eat our way to health, but it doesn't seem to work. We can eat our way to general health, but exercise and sleep and stress reduction. Cortisone, which is produced by the brain and elsewhere, is a stressor, and it will influence the immune response. It will lower it because cortisone is an anti-inflammatory, among other things. And we use it when we're in the episode of almost an inflammatory response. We release cortisone to try to reduce the inflammation. So we have taken it and used it both as a topical, for itches and so on, but also as prednisone type. Now, from there, I just want to jump to one of the greatest killers in history, smallpox, a variola virus of the pox virus group family. It's an orthopox virus. It's inhaled usually or direct contact, has a slow incubation, and it's highly infectious even when you're incubating. This particular one is only in humans, and there's 2 variants, and you notice that one is very minor, less than 1% death rate, but the other one is 30% death rate, or third. And this also is a very scarring condition. It's mostly respiratory. You'll fever initially in that first week. And then after the prodrome stage, you will develop early rashes, which will be maculary. And then these will turn into after 3 weeks, you'll turn into papillary. Papillary, then these will blister and vacuolate and then turn into these pustules, which will actually flatten out, which are called flat pustules. This is the malaise and the fever, and if it gets to this stage, it's going to be very, very iffy. Like a third of the people will die, but also you will be left completely scarred. Various meant spotted in the Latin, and so it was used to show the spotting of the pustules and so on. And then it was called smallpox to distinguish it from the great pox, which was syphilis. And syphilis can cause pox-like skin eruptions as well. So it was not known which caused which. And of course, chicken pox, which is a much milder case, and they don't have the same pustular development. There are others that affect us. We basically can get monkeypox. We can also get cowpox, which turned in, or a type of vaccinia cowpox, and another cowpox, which can travel between rodents and cats and cattle and eventually on humans. There are many others that attack monkeys or cattle, and they all have related etiologies and pathologies. So they basically attack everything: camels, pigs, rodents, and of course, this is swine. And the relationship is pretty well known that cowpox and variola major are very, very closely associated. And because of that, Jenner was able to use cowpox and inject pustules of cowpox into people to protect them from major smallpox. There are monkeypox, which we can catch, camelpox, which we can catch, and camelpox actually gave rise to this other form of cowpox. We know a bit about its history. We don't know exactly when it came out of Africa, but I suspect, and other people suspect, that this was maybe 50,000 years ago when modern humans came out of Africa and perhaps were carriers. So when we say thousands, I think this was 40,000 years ago, probably infected Neanderthals. There's no evidence, because we don't really have much evidence that there would be. And then from this, it came to mice or gerbils, and then it went to camels. And then it worked its way throughout Europe and then over into Asia. And we know then when it spread. Recently, it was discovered in Viking burials in 1700s. Actually earlier, about 1000 AD, that smallpox made it up into the Scandinavian regions. So we can look at the genetics and know where these things came and how they spread. We also know that here's King Ramses. He is highly pockmarked, and we know when it spread across Europe and when it varied. So it wasn't that long ago that it may have jumped from the mice to camels and then to humans. That's not unlikely because MERS, very like our current COVID, is derived from the Middle Eastern respiratory syndrome, is actually derived from camels. So another zoonotic contributor. We know from looking at corpses when smallpox hit various populations, and smallpox had wiped out not only native populations all over the world, but also resident populations. Smallpox was very destructive in Boston. Now, a couple of quick stories. Lady Mary Wortley, born into royalty, was known as a beauty, was popular in the court. And she married a diplomat and went to Istanbul. And there she witnessed some of the first, what were then called inoculations. And this wasn't the first time of inoculation. The Chinese did this at least 1,000 years before. That is, they just took a needle and poked it into smallpox and pustules, and then poked the person. So she had her young son, Edward, who was 4 years old, done in Istanbul while she and her doctor watched the process. Later, she came back to England and told people about this, wrote about it, and then she convinced the royal doctors to come and watch as her personal physician, Dr. Maitland, did the same inoculation to her daughter. And over the time period, a couple of weeks, all these royal surgeons would gather and watch the recovery of their daughter. And since she was so close to Prince Caroline, she convinced her the Princess of England to do this to her children, but not until the princess decided to inoculate Newgate prisoners with the promise of freedom, and which they did, and then she inoculated her daughter, 2 daughters. After that, following the royal privilege, people flocked to get inoculated. This is in early 1720. And not until almost 30 years later did Edward Jenner, who was a practicing physician in Gloucestershire, notice that milkmaids often had minor cowpox, but remained unscarred. And smallpox is very, very scarring, hideous. Maybe you've seen a cowpox or chickenpox vaccine or pox on the skin, but smallpox was debilitating. So he took pustules and he dipped a needle in it and then inoculated, or in this case, he called it vaccination after the cow, vaca. And he called this process vaccination. And the cowpox was close enough to the smallpox that once the cowpox grew, it protected the children from further disease. Now, of course, nothing is new. People are people. So there was an amazing anti-vaccination hysteria. And of course, people began to envision cows coming out of their body because they didn't think it was Christian to put anything foreign in their body. So you could run this cartoon today and understand what it meant. But eventually it became compulsory, and even with inspectors, and remarkably, Jenner's vaccination became very, very effective and nearly disappeared. The King of Spain had heard about this, and he got orphans. Well, he had heard about the inoculation from Istanbul. It had worked its way across Europe. And so he put together a fleet with 20 orphans, and he infected the first orphan with smallpox. And they went to the Caribbean and South America, and they used that orphan to inoculate everyone there. And then they inoculated another orphan and went to the Pacific coast of South America. And then they inoculated another orphan. And they kept using these feeders to inoculate Spanish citizens all over the world. They went to the Philippines, and they went to India and elsewhere. Interesting story. I will refresh you about it the next time, but it's fascinating. The World Health Organization, 1959, smallpox was beginning to decline. They undertook a worldwide inoculation and concentrated on different countries, of course, the European and American countries in the 50s, and then later all the way into India, and the last case was noted in 1980. Actually, it wasn't the last case because someone contracted it in a laboratory situation. But smallpox, thanks to the World Health Organization, is no more, except two types of smallpox are kept as weaponized versions, some of them chimeric, that is mixed with other viruses. Recently, a year ago, almost exactly, one of these facilities, the Vector Research Facility in Siberia, Novosibirsk, blew up. They claim nothing was released. The other repository for this is in Atlanta, Georgia. And if you're in the military, you still get smallpox vaccination because we know that it's around and potentially usable as a bioterroristic weapon. The United States military has run a number of different exercises. You've probably never heard of these. One in 2001, one in 2015, called Dark Winter. This one was a weaponized smallpox, not real, but they used a dust and a mist and would release it and watch where the dust went, or release it into buildings. This was a VPOX. It was a Venezuelan encephalitis hybrid pox virus. And then they estimated what the kill rate would be. Fascinating stuff if you're interested in stories. We have many other things. We have Marburg virus and a bunch of other things, monkeypox, and a whole bunch of nasty toxins from bacteria that are weaponized. We didn't start this, actually the Japanese did, and the Koreans, and well, we did. We used smallpox infected blankets and gave them to the American Indians to wipe them out. But that's another story. Now, vaccines. With Lady Montagu and Jenner, vaccination became possible. Pasteur developed a series of vaccinations for rabies, which is remarkable because it's such a difficult disease. But look at the decline in cases of, say, just measles or whooping cough or mumps. And when we show this kind of thing, sometimes people show that, oh, look, this is kind of before vaccinations and already it was declining. Well, this is the role of sanitation and toilets and clean drinking water. But they like to say the death rate was declining even before vaccinations, but you got to read the whole graph. So different vaccines were introduced at different times. And what is a vaccine? Well, it's to prevent. It can give you immunity either actively or passively. You can actually weaken a live vaccine or a live virus. That was problematic with polio because sometimes polio would revert. And so this is not a preferred strategy. You can attenuate it and try to inactivate it even more. That also can revert. So what you try to do is figure out subunits where you can just say, get the protein markers from the surface, or you can combine different things. You can just take the DNA out of the virus and put it into another host. You can make synthetic virus particles that are not going to kill you but have the same surface markers. And there's a whole host of others. But there is, we do still use oral polio, which is live attenuated, measles, rotavirus, yellow fever, and smallpox. These were all attenuated, meaning weakened. And there are others if you want to look at these. Weakened virus, you use formalin or X-rays or radiation to weaken it so that once it gets in, there'll be a good immune response and you'll get all kinds of antibodies produced. And there is a danger of those being regressing in a sense. If you take the gene out of the coronavirus that makes the spikes, you can put it into, say, a lentivirus or an adenovirus that's going to be harmless. But that gene is there, and if it gets eaten by a dendrocyte or a macrophage, it will present every one of the antigens. And so there's the spike, and it will then activate the T cells and the B cells and so on. That's a very effective, simple way of doing it.